Hello, biologists. Welcome to lecture number 1 of BI212. And today we are going to talk about the structure of DNA. So first, I am showing you a nucleotide. This whole thing on the left here is a nucleotide. And a nucleotide is composed of 3 parts. First, the phosphate group, which is highlighted in yellow. Then the 5-carbon sugar, also called a pentose sugar because it's a pentose shape, and then a nitrogenous base. So the phosphate group and the 5-carbon sugar are the same in all of our nucleotides, but the nitrogenous base is different in our different types of nucleotides. We have 5 nucleotides. You can see them over here. Here are your 5 types of nucleotides: guanine, cytosine, adenine, thymine. Those 4 are found in DNA. And if we're looking at RNA, you're going to use uracil instead of thymine. Each type of nucleotide only pairs to one other nucleotide. So guanine always pairs to cytosine. And I remember that because G's and C's look really similar to me. And adenine always pairs to thymine. And I remember that because it spells at. So at, A and T always go together, unless you're looking at RNA, and then A and U would go together. Uracil. Here, looking back at our large nucleotide on the left, I want to talk about the carbons in our 5-carbon sugar. So at the top of our pentose 5-carbon sugar, we have an oxygen molecule. And then if you go clockwise around this shape, we're going to count off the carbons. So clockwise, the first one is 1. And notice that they're not showing you the C. It's just assume that when you have a junction like this, there's a carbon there. So you could assume there's a carbon here as well and a carbon here as well. So this is our first carbon, even though it's not being shown. This is our second carbon, our third carbon, our fourth carbon, and then outside of our pentose shape, your 5th carbon is right here, and that one is being shown. So that's our 5th carbon. So the phosphate attaches to the 5th carbon. And when we have another nucleotide that's going to join this one to make up a backbone of DNA, the phosphate group would be down here, and it would bind to the 3rd carbon. So this becomes important because when we look at the overall structure of DNA, we say that it is anti-parallel. Let me show you what that means. So here's a double strand of DNA, and it's going in 2 opposite directions in respect to each other. So it's the anti-opposite. And then parallel because they're parallel to each other, just going in opposite directions. If we look at this nucleotide, phosphate, sugar, nitrogenous base, so this is one nucleotide here. It has the oxygen on top, and it has the carbons numbered 1, 2, 3, 4, and then outside the 5th one right there. Then, attached to this 3rd, with a phosphodiester bond, we have the phosphate of the nucleotide below it. So this is one nucleotide. And then attached to this phosphodiester bond at the 3rd carbon is the nucleotide below it. So this strand is running from 5 because 5 carbon is on top to 3 because the 3rd carbon is on the bottom. So 5' to 3', which tells us that the 5' is on top and the 3', this 3rd carbon here, because we're counting clockwise from 0, 1, 2, 3, is on bottom. If we look at our other side of DNA, note that the oxygen's now pointing down. So this is upside down compared to the side on the left. So if we count off our carbons again, going clockwise from the oxygen, it's 1, 2, 3. So now the 3rd carbon is on top, and the 5th carbon here is on bottom. So this side is going from 3' on top to 5' on bottom. So in different directions, anti-parallel. That's why we need to count the carbons, because we need to know what direction our DNA strands are oriented in. Here we can see the hydrogen bonds between our nitrogenous bases, so adenine, thymine, at, and cytosine, guanine. It also labels our 2 different types of bases, so pyrimines or purines, and that's there. So if we look at the 3D structure of our DNA, so we're kind of looking at it flattened out right now, so it looks like it's not spiraling. But if we look at the 3D structure here, it's been flattened out, but then it's allowed to spiral. You can see the double helix shape. So double being 2 strands, and helix because that's like a spiral staircase shape. So each one of those A's and T's on the little green bar and this bit of the strip up the side is a nucleotide. So this is 1 nucleotide, 2 nucleotides, 3 nucleotides, and you can see that the base pairs are hydrogen bound together as expected. The C's and G's are together and the A's and T's are together. So in each one of your cells, you have 6 feet of DNA, and that's really easy to get like tangled up, right? So you want to be able to compact it and organize it for when you do mitosis, which you're doing all the time. So to compact it, what you're going to do is you're going to take your DNA and you're going to wrap it around some special proteins called histones. So histone proteins, wrap some DNA around them, kind of like you would wind a thread around a spool or wind your earphones up around that little thing that they come with. And once you have DNA wrapped around histone proteins, you call it a nucleosome. Your nucleosomes are going to get wrapped up even tighter, and those little nucleosomes organize into these little complexes, which eventually condense into chromosomes. So a chromosome is a whole bunch of DNA that's wrapped around histone proteins and condensed together so it's easy for you to move around your cell during mitosis. And that's why it becomes visible in mitosis. Remember, it becomes visible during prophase, and why it kind of isn't visible the rest of the time, because you want to have your DNA open to access it unless you're trying to move it around. So chromosomes are compact pieces of DNA that you can move around and do things with. This image also is highlighting a gene. So a gene is a piece of DNA from here to here that you're going to use to make a protein. Whatever that protein is. Maybe you're going to make histones, maybe you're going to make ribosomes, maybe you're going to make muscle cells, whatever it is, because we use proteins to do like everything inside of our cells. So a gene is a piece of DNA used to make a protein, and your DNA is made out of nucleotides, which have those 3 components that you just looked at. So this is already cool, right? But we didn't know any of this until like the 50s. And I want to talk about the discovery of the shape of DNA because it's a really interesting story. And it's a story that highlights some of our issues in the sciences that we need to talk about more. So who discovered the structure of DNA? Do you already know the answer? Well, the 3 people that got the Nobel Prize for discovering the structure of DNA are Francis Crick, James Watson, and Maurice Wilkinson. And you can see at the label at the top, let's talk a bit about institutionalized sexism and racism. It's important to talk about how these things are involved in the story of the structured DNA and what actually happens. So these 3 guys get credit, but some of the credit should also be shared with a woman named Rosalind Franklin. So Rosalind Franklin was a female Jewish scientist, and she was working at a different university than Watson and Crick. And Watson and Crick were working with models, kind of like erector sets, if y'all know what those are. And they were trying to model the shape of DNA, but they hadn't figured it out quite yet. And at her university, Rosalind Franklin was using X-ray crystallography, which is where you take a little item like a protein or DNA and you freeze it in a crystal and you shoot X-rays at it. And based on how the X-rays bounce off, you can see the shape of the thing inside the crystal. So she was working on the structured DNA on her own, separate from them. Rosalind Franklin's work was shown to Francis and Crick without her knowledge. And Watson has this great quote that when he saw the picture that's up here in the right, it's called photo 51, his mouth dropped open, his heart started beating, he immediately knew what the structure was, and then him and Crick go back to their lab and they work it out and they publish it before Rosalind Franklin. So it was her work that gave them the inspiration for their work, and they were just quicker at getting it out to the public. He also has quotes saying that in a few weeks, she probably would have published it herself, and then she would have gotten full credit. Now, Rosalind Franklin didn't know that they had looked at her work. And Unfortunately, she didn't share any of the credit in this because by the time those three men were awarded the Nobel Prize, Rosalind Franklin had died of ovarian cancer. She died at only 38. So she died only 5 years after taking this photo. And we do not award Nobel Prizes posthumously. So she was not included. So the reason this story is really interesting is because it highlights kind of the institutionalized sexism and chauvinism that is really common in the sciences historically. Like, it was designed for males, and it was mostly males that were conducting the science. And many of the males are problematic. James Watson, the one who got the Nobel Prize and who is often put on a pedestal in a lot of textbooks and a lot of websites and anywhere where you find information about his contribution to the sciences, is especially problematic. And we need to be talking about institutionalized sexism and racism because we can't learn from our mistakes. And I'm saying "are" as like science as a collective until we identify what the problems are. So I recommend reading this article. I'm not showing you the whole article because for me, it's really upsetting to read the things that he read or that he wrote or that he said. The title is DNA Scientist James Watson Has a Remarkably Long History of Sexist, Racist Public Comments. So you can check it out yourself. There's a whole lot of offensive things in there, so please prepare yourself for that. And then in 2019, so pretty recently, James Watson says this really horrible thing. Go ahead and read it. It's on the bottom there. And it's horrible for many reasons. First of all, it's just wrong. So there is no intelligence difference based on race. That's just incorrect. It's offensive. It's ignorant. Like, it's just wrong. And this is a person who our group of folks, like scientists, have held up and said is an amazing person we all should be trying to emulate. And so it's extra damaging when a person like that says these really racist, incorrect, offensive things. So after he said that, he was stripped of all his titles. There's some really good headlines here, like he may have unraveled DNA, but James Watson deserves to be shunned. So a nice DNA pun there for you, because unraveling. And after this, he's pretty much not talked about anymore. And it's interesting because we need to be highlighting when things are wrong, and we need to be learning how to make things right. So it is appropriate, in my opinion, that he loses his titles, and that now we talk about him as like an example of what should not be celebrated in the sciences. Because we in this class, we know that science is for everyone, and we don't need racist, sexist, homophobic, anti-Semitic people setting guidelines that we're supposed to be emulating. I do want to move a little bit further, and I want you to think about this practical question. Is institutionalized racism and sexism still a problem in the sciences? So you read those options, choose the best one. If you said any of them, you are correct. And it is still a problem. It is still a problem, and we still need to be talking about it. And we still need to be figuring out what we can be doing to make it better. And to help us figure out how to make it better, I have some resources for us. A lot of these are from 2020 when we were talking about racism a lot in America. And I think that's a really important thing to be doing, and we need to be doing it continually. We don't get to just stop talking about it. Time magazine has these 3 steps to combat racism. So first ones have empathy, stand against rhetoric that reinforces stereotypes. That means highlighting when people are doing things wrong, like James Watson, and talking about what's wrong so that we don't just sweep it under the rug. And then the third is being an active consumer of media and news. By active, they don't mean a lot, they mean being critical. So that means using the CRAP tests like we learned last term to evaluate what you're reading, to figure out whether or not it's biased, to figure out whether or not it's trying to sway your opinion, and then deciding if you should be internalizing it or not. Excuse me. We also have a couple more things. This here, the Medium article, is 103 things white people can do for racial justice. And if you're not white, it's still useful. It's not just for white people. It's 103 things anyone can do, but it is geared toward white people, because being a white person means that you're often unaware of what's going on. And so you need to work extra hard to educate yourself, especially if you're in a place of control, like possibly a teacher, which is why I'm doing this. We need to be working together on this. We also have how to be anti-racist and how to speak out in your own circles from CNN. So 2 good resources. And then the way I learn is by reading. So I have a bunch of book lists for you. So here are 22 books on race and white privilege. We have 7 anti-racist books. We have 20 books for 2020. And then if you're not a big book reader, which I know, especially during the term, you might be too busy to read on your own, I also have films and podcasts that you can be checking out. So a bunch of resources so we can start to think about how to make the sciences less racist and less sexist. And one of the things that we can be doing, and to relate back to our content, is to be learning about the people who added contributions but maybe didn't get the credit. So there is a documentary from Nova. It's called The Secret of Photo 51. That's that photo I showed you earlier. And it's all about Rosalind Franklin and how she discovered her amazing contributions to science. She was also really critical in figuring out the shape of the polio virus, so pretty neat. And if you go to this website here, the PBS website, that's the official website, and you have to pay to watch the documentary. Totally worth it, highly recommended, but I don't want the payment to be a barrier, so I included 2 other websites below that you can watch it at for free. All right, that's the story of the structure of DNA and a little bit of how to work through some of our problems in the sciences. And if you have any questions, email me. I'll talk to you soon.